Hello everyone, so welcome to our focus on glycolysis, and this is part 1 of 3, and again we're going to start with glycolysis, move the citric acid cycle, then electron transfer chain, move our way down the cellular respiration, but yeah, again this is part 1 of glycolysis. So this is kind of our big overview picture with little detail, but just kind of the overall process. So this is the whole purpose of metabolism, is ATP production, as we mentioned in biology. So we have the intake of carbohydrates through food, either via plants that contain glucose or animals that have consumed plants. Then we have our glucose, which enters glycolysis. This glycolysis produces the pyruvates, and these pyruvates form acetyl-CoA, and acetyl-CoA enters, but fat can also become acetyl-CoA, but we'll talk about that later. Will enter the citric acid cycle, and again, proteins can also become amino acids. And so the citric acid cycle goes, then we lose 2 carbons. Carbon dioxides are produced, which need to be eliminated as waste. Through the electron transfer chain, we form our NADH, FADH, and different molecules that enter the electron transport chain that transport electrons. Then in the electron transfer chain, we have energy released in several stages and the production of lots of ATP. So first and foremost, essentially glycolysis is considered glucose oxidation in a sense. But this is kind of, glucose oxidation is the overall process essentially of using glucose and being oxidized to gain energy. So oxidation, so essentially our usual definition is the loss of electrons, as we mentioned, but the term also means interaction with oxygen with other substances. So literally oxidation with oxygen. So the oxidation of glucose is an oxygen requiring process. So this is why we need to breathe in oxygen for this energy metabolism to occur. But although not all stages involve reaction to oxygen. So throughout this entire cycle here, our input is 6 oxygens and glucose and 38 ADP. So that's our diphosphate. The output is 38 ATP, 6 carbon dioxide, which needs to be expired, and 6 H2O. And again, the breakdown of glucose to release energy as ATP, that is the goal. So, again, this glucose oxidation is essentially aka called cellular respiration, which you've probably heard me say. So again, this is essentially synonymous with glucose oxidation. So process organisms undergo, so essentially cellular respiration is a process organisms undergo that converts glucose to energy, usually using oxygen and glucose to produce ATP, CO2, and water, which we saw as our products down here. ATP, CO2, and water. The broader definition, cellular respiration is a broader definition than glucose oxidation since it involves all food that can be used to yield ATP, including fatty and amino acids. So there are scenarios where fatty acids and amino acids can enter these cycles, and it's not 100% only glucose. So the phases of glucose oxidation or cellular respiration is 1, glycolysis, which occurs in the cytoplasm. So this part does not need oxygen. Then we can have pyruvate dehydrogenase reaction. So remember our dehydrogenases reaction, which involves the transport of pyruvate into the mitochondria from the cytoplasm. Then we have our citric acid cycle or a Krebs cycle or tricarboxylic acid cycle, which occurs in the mitochondrial matrix. And then we also have our electron transfer chain, also seen as ETS or ATC, sorry, and occurs within the inner mitochondrial membrane. So again, you can see our inner membrane, outer membrane, and matrix. So again, here's the mitochondria, and this is kind of a picture that depicts, that shows where each happens. So we have our glycolysis, like we said, which occurs in the plasma, cytoplasm, the cytosol. Then we have our pyruvic acid, then we have pyruvate dehydrogenase occurring. Then we have a Krebs cycle occurring in the mitochondria. Then we also have electron transport chain and oxidative phosphorylation. So this is a kind of a good summary figure to get all the more broad in and outs of it, but at least shows you the net gain and loss of whatever happens in each stage. So again, we have their stages 1, 2, and 3. And stage 3 and 2. Stage 1, 3, and 2, sorry. And then, so again, glycolysis in the cytoplasm, electron transfer chain in the mitochondrial membrane, citric acid cycle in the mitochondrial matrix. So here we see our glucose becomes 2 pyruvate molecules, and we produce one NADH and 2 ATP. Then we have shuttle transfer reaction equivalents. We have our NADH and FADH. These are all electron transport, which are you can see here that NADH and FADH from all sources donate electrons to the electron transport chain. And these come from here, as well as here, from the electron transfer chain. So here our pyruvate, we have the pyruvate dehydrogenase reaction. We form our acetyl-CoA and a CO2, as well as NADH. But again, there's also things like fermentation reactions, which we can talk about if we don't have oxygen present, which we talked about in biology if you want to check that out. Then we have a citric acid cycle occurring. Then we produce this, and these go back into the citric electron transport chain. And then we continue with oxidative phosphorylation. And electron transfer occurs via a series of electron carriers, forming lots of ATP. But again, we'll move through these in a lot of detail over the next lectures. Again, we have 3 lectures on glycolysis, and then we'll move on to the other stages. So glucose uptake. So glucose cannot diffuse directly into the cell, but enters via its own transports. 2 transports. So glucose can enter cells via 1, facilitated transport via transporters, or 2, co-transport with sodium. So glucose cannot enter the cell without aid because it is a polar molecule. It has 2 different charges. So as we talked about, we have our cell membrane with our phospholipids, with the hydrophobic and hydrophilic areas, and due to its polarity, it cannot enter. So it needs a transporter or to be co-transported. So facilitated glucose transport. So facilitated glucose transport is mediated by a family of at least 5 glucose transporters in the cell membrane designated as GLUT1 to GLUT5. So there's a lot of talk about GLUT4, or glucose transporter 4, in exercise, and how exercise can stimulate the transport of glucose into cells, lowering blood sugar and stuff like that. So this is kind of the primary principle why exercise is good for people with diabetes and stuff like that. So GLUT4. So GLUT4 is a unique transporter that is located in the cytoplasm. GLUT4 translocates to the plasma cell/cell membrane when glucose disposal is required. When glucose disposal is required, this occurs mainly after eating, so we have that spike in glucose after eating food. So GLUT4 transporters know they have to move to the membrane due to insulin. So insulin is essentially the signaling hormone for GLUT4. So again, steps involved in disposal after meal. So we eat that meal, and we have that glucose spike. So first, we eat the meal. Then glucose from the meal is absorbed into the circulation or the bloodstream. The pancreas, so our pancreatic beta cells, and the islets of Langerhans detect the increase in blood glucose and begin to secrete insulin. So our beta cells secrete insulin while alpha cells secrete glucagon, but we'll get to that later. So insulin travels through the bloodstream to the muscles, fat, and heart. Insulin binds to insulin receptors on the plasma membrane, and this recruits GLUT4 transporters to the plasma membrane. Glucose enters the cell, and cells, and blood glucose is maintained within a narrow range. So note, in skeletal muscle, there are 2 distinct pools of GLUT4 transporters. One pool is exclusively stimulated by insulin, and the other pool is exclusively stimulated by muscle contraction. So essentially, the mechanical stimulation of muscle contractions can activate these GLUT4 transporters, or GLUT4 receptors, and increase glucose uptake in an insulin-independent manner. So this is essentially a way, essentially, if you're a type 1 diabetic where you don't have any insulin, you can actually lower your blood sugar quite a bit with exercise. And this is why certain type 1 diabetics need essentially to monitor their sugar while they're exercising, because they become hypoglycemic when they exercise because they already have issue with insulin because they don't produce it. So the blood sugar can get high, but they can actually get quite low as well when they exercise. So they have to monitor it very carefully. So essentially, there's a little cartoony graphic of insulin-stimulated glucose uptake. So you have our insulin, binds to our insulin receptor. Here's our glucose in the blood. So this is our blood vessel. because again, it's a hormone, can travel in the blood. Then here's our GLUT4 receptors. So essentially, insulin receptor gets stimulated. They stimulate the GLUT4 receptors within the cell. They move to the cell membrane and then allow glucose to pass through. So stimulation of non-insulin mediated dependent glucose uptake and contraction mediated glucose. So again, we have contraction of our muscles occurring. This results in an increase in adenosine monophosphate to ATP ratio. So this is when your body detects this AMP versus ATP, it's eventually saying, oh, we're low on energy because this is a non-energized form. Then this is called AMPK, so this adenosine monophosphate kinase. And again, we see our kinase. We know kinase involve phosphorylation. So see AMP activated protein kinase, sorry. And then, so this activates our GLUT4 again, and then boom, go up, GLUT4 activating, letting glucose come. So again, I messed this up. AMPK is AMP, so adenosine monophosphate activated protein kinase. So remember, we said the second mechanism. So these are facilitated through receptors, and this is the co-transport mechanism we discussed. So we talked about the sodium glucose co-transport mechanism. So this is an energy requiring process. So again, energy requiring means ATP. Here we can see ATP becoming ATP inorganic phosphate that transfers glucose against this concentration. So that's where we can say it requires ATP because it's moving against its gradient. So if it was just moving with its gradient, it could usually be transported through facilitated diffusion, but this is an energy requiring process. So essentially, this is what is considered a symport mechanism. So we have our sodium, potassium, ATPase pumps, which are functioning all the time. So essentially, these are working all the time, pumping sodium out and potassium in, using energy. So essentially, there is a concentration gradient of sodium here because sodium is constantly being removed from the cell due to sodium, potassium, ATPase. So as the sodium decreases because of ATPase, there's a low concentration but a high concentration outside. Sodium will move in based on its concentration gradient, and glucose will essentially tag along, or they're synergized essentially, and will move with the with sodium. But again, it only works when this ATP is active, pumping other stuff out. So it hops on another concentrations gradient. So again, a symport mechanism. In the symport mechanism, glucose couples with sodium to enter the cell. Glucose is moving against its concentration gradient, and it results in the use of ATP. So the fate of blood glucose. So again, glucose is in the blood, so here's a little glucose. Here's our cell membrane. So as it moves into the cell, glucose can either become glycogen, starch, or sucrose, essentially becoming stored. So you might have heard of muscle glycogen or liver glycogen. So your glucose can be stored for later use. So essentially, this is like our energy reserves. So a lot of like kin stuff, you'll hear about muscle glycogen, how that when you're exercising, this is that stored energy you have in your muscles that can be burned prior to essentially using your other types of storage materials. Or the glucose can be directly used through oxidation via glycolysis, forming that pyruvate, and then moving into the other pathways of citric acid cycle and electron transport chain. So glycolysis. So glycolysis occurs constantly in every cell, and the rate of glycolysis changes depending on the demand for ATP and glucose available. Glycolysis begins with glucose and ends with 2 pyruvate. So every 1 glucose gets 2 pyruvate. So when we move through the citric acid cycle and stuff like that, and electron transfer chain, we can essentially say that it's all happening at the same time. So this will technically happen times 2 because there's 2 pyruvates. But a lot of the times it's depicted as one pathway, but you have to realize that it's happening actually kind of twice because there's 2 pyruvates that when basically most of the times when we talk about a citric acid cycle, it's describing it with 1 pyruvate molecule. So essentially we have our glucose plus 2 ADP plus 2 NAD+. So that's essentially, these are all the constituents of glycolysis, form 2 pyruvates and 4 ATP and 2 NADH. So this NADH is important because it enters, you need it for other passages. And again, same, this is our initial ATP. So if we never had oxygen, no O2, this is where we'd stop essentially. And then this pyruvate would enter fermentation or form lactic acid or alcohol, but we'll go back to biology if you want to look at that. So again, this NADH is involved in oxidative phosphorylation. And the overall Gibbs free energy, or Gibbs standard free energy of glycolysis is negative 19 kilojoules per mole. It is a spontaneous and irreversible reaction. So once that glucose is broken down in that 2 pyruvate, it can't go back. So the 2 major functions of glycolysis are 1, to generate ATP in the form of 2 ATP. So here's that 2 ATP. To generate pyruvate for final oxidation in the mitochondria along with NADH. So again, the overall standard free energy of glycolysis is negative 19 kg per mole. kJ, sorry. So types of reaction in glycolysis. So these are the ones we mentioned in a very short lecture. So in glycolysis, we have hydration and dehydration reactions, which involves the addition and removal of H2O. And these involve hydratase and dehydratase enzymes. We also have oxidation and reduction. So these become quite important because these electrons are key in glycolysis because we need them for the electron transport chain. But we'll talk about that later. So again, oxidation reduction involves the loss and gain of electrons. And these involve our dehydrogenase enzymes. And hydrogenase enzymes include NAD plus and FAD coenzymes, which we'll see and are quite important later. We also have phosphorylation and dephosphorylation. So again, our kinases, which is the loss and gain of inorganic phosphate, which is that PI. And these involve kinase or phosphatase enzymes. So here's the very, very big picture overview. So we go from that 1. Also, just want to mention quickly that it's very important once we start moving through these pathways to count the carbons. So counting the carbons is quite helpful of knowing what stages are occurring at what time. So know that we start with 6 carbons and end with 2 3-carbon molecules. So that's important. So again, we go from that 1 glucose. There's 2 reactions where we form energy, we get that 2 ATP, and we form 2 NADH. And then throughout this, we get our 2 pyruvates, 2 pyruvate molecules. So again, 6-carbon glucose is split into 2 molecules of 3-carbon pyruvate. ATP is consumed, but more ATP is produced. So we need some ATP to go into this in order to actually gain this ATP. And we also have NADH being produced. So here's a much more detailed look over here. So this essentially covers most of the reactions. Well, all the reactions this is for 1 pyruvate. So again, this is only showing the 1 pyruvate produced. So again, we go from our glucose. Again, we're going to go through each of these steps in detail, looking at each enzyme, each product and reactant, and especially focus on when energy is used, when energy is gained, and then when these certain things like NADH are produced. So I say the main things to focus on are what stages energy is used, gained, or when we have our reduction reactions or oxidation reactions, because those have long-term effects in the rest of the chain. So again, glycolysis starts with glucose. There's this 10-step process, and there are 3 main stages, which we'll talk about soon. Each step uses a different enzyme and has a different reactant and product. It's not overly important to remember each enzyme, but it's good to remember this kind of part to actually understand what's happening between this phase and this phase. Not necessarily the name, but also knowing the name does help in some sense that it tells you what's happening. But it's just important so you actually know when you see this, even if you don't memorize it, you know what's actually happening between biphosphoglycerate and phosphoglycerate, 3-phosphoglycerate, essentially due to what you're being told. So remember, kinase involves phosphates, for example. So again, the product of one step within this cycle becomes a reactant of the next step. So essentially a pathway where this molecule from here changes to this molecule, and this molecule changes into another molecule, etc. Again, it's the splitting of glucose, which is a metabolic process, and the pathways of glucose splitting is a metabolic pathway. So this is a really good study tool of the overall, like, essentially the main important things to take away, especially if you're doing a more like this intro type biochemistry where you're not, going into extreme detail. So as we mentioned, there's those 3 stages of glycolysis. So again, the first stage is the priming stage. So in the priming stage, this is the number of steps, by the way. So priming stage, we have our glucose 6-phosphate to fructose 6-phosphate. Fructose 6-phosphate, we have ATP, energy went in, and fructose 1,6-biphosphate. And then here are the important enzymes. So this is one that's talked about quite a bit, is phosphofructokinase, which you'll see a lot of the times PFK. But anyways, we'll get to that later. The second stage is the splitting stage. So you can kind of get where we're coming. So priming is kind of like, we're putting energy in. You see the ATP goes in. The splitting stage is when we go from that glucose to the to the splitting. So the glucose is now split. So here we can see we'll see later that we get our dihydroxyacetone phosphate as well as our glycerol-aldehyde 3-phosphate. Then the third stage is the oxidizing and reducing reaction. So this is when we're going to see our agents here, oxidizing and reducing agents, NADPH, NADDH, NAD+, etc. And then we'll see all these different important enzymes. So again, we recognize our kinases and our dehydrogenase. So that's our oxidation reactions. So how some reactions in glycolysis have a negative change of free energy while others have a positive. So we can see here that certain reactions are very large, and this red is irreversible. So that red is irreversible, and you can tell by the arrow. So that would be an arrow without this, and then reversible would have an arrow that's like that, which you can see here. So that would be reversible. This is irreversible. So just keep that in mind when you're looking at the figure. So again, we have some large positives as well as some negatives. So remember, actually it has more. So overall glycolysis, we talked about, had that overall charge, and it has a negative change of free energy, which is different from the standard free energy. So remember that in biology, the conditions are not standard. What determines whether a reaction really occurs is the free energy at that particular moment. But the change of free energy and the standard free energy usually, but not always, correlate with one another. So if the Gibbs standard free energy and Gibbs energy usually correlate, that still leaves us with a question. How can there be reactions with a positive change in energy in a metabolic pathway? So there's 2 explanations. So one is that they're not exactly the same, and the Gibbs free energy, that must be negative for a reaction to occur. Although the 2 correlate well, the reactions with a small positive Gibbs standard free energy can be made to have a negative energy by small changes in the concentration of reactants and ATP coupling, which you mentioned before, which puts energy into the reaction and can change the sign of the standard free energy from positive to negative. So Gibbs standard reaction, free energy, and reversibility. So reactions with a small change of free energy are likely to be reversible, while reactions with a large, either positive or negative, are likely to be irreversible. There are 2 exceptions which are reversible. This includes aldolase, which is plus 23, and 3-phosphokinase, phosphate kinase. So here, even though it's a large number, it is still reversible, and this is also for 3-phosphoglycerate kinase. So again, they're big, but they are reversible in comparison to here with phosphofructokinase, that's irreversible. Same with hexokinase. So now we're going to move through each step of glycolysis slowly. We won't cover all steps in this lecture, but we'll slowly move through it. So step 1 of glycolysis is the addition of energy is a regulation step. This is irreversible with a large change of free energy. So glucose, with the addition of ATP, with that phosphate becomes a glucose 6-phosphate. So this 6 indicates the carbon. So this is essentially carbon 6 there's a phosphate being added to carbon 6. So again, our substrate is glucose, the product glucose 6-phosphate. The biochemical process is phosphorylation. We're adding that phosphate. Again, this uses kinase, so this one was hexokinase. And again, a possible name can be considered glucose kinase if you want to be technical, but the name is hexokinase. So again, it's the addition of energy. So glucose plus ATP equals glucose 6-phosphate plus ADP. This is considered glucokinase when it occurs in the liver, and hexokinase when it occurs in any other tissue. So this change of free energy is negative 16.7. Again, this is a large negative gives standard free energy, therefore it is spontaneous and irreversible reaction. But we mentioned there was those two exceptions. Step 2 of glycolysis. Now we go from glucose 6-phosphate to fructose 6-phosphate. So we can see here we're having a change from glucose to fructose. So as we mentioned before, isomerases involve changing in the geometrical structure of a molecule. So we talked about glucose to fructose being just a change in structure, nothing else. So again, this is why this is an isomerase and involves isomerization of glucose 6-phosphate into fructose 6-phosphate. So again, substrate is glucose 6-phosphate, the product is fructose 6-phosphate. Biochemical process isomerization using an isomerase. The proper enzyme name is phosphohexokinase isomerase. So important, one thing to point out. So even though we started at step 1 from glucose to glucose 6-phosphate, is possible for storage versions of glucose as glycogen. Glycogen can enter at glucose 6-phosphate. This is important because it actually requires less ATP on our part. See here, we needed ATP. When we use glycogen in glycolysis, it enters straight at glucose 6-phosphate, so we save 1 ATP. But we'll talk about that more later. So again, continue on step 2. Glucose 6-phosphate with a small reversible gives free energy to fructose 6-phosphate with phosphohexose isomerase. So both glucose 6-phosphate and fructose 6-phosphate are phosphorylated hexose sugars, so both could be named phosphohexose. Again, a small positive energy, therefore it is spontaneous but reversible reaction. We can see that with the arrows. But it's also good to understand why those arrows are there. So step 3. Step 3 is fructose 6-phosphate. So go fructose 6-phosphate to our fructose 1,6-biphosphate. So again, a lot of these numbers refer to the phosphate location. So here we can see bi is essentially 2 phosphates. So we can see another ATP is going in and another kinase or phosphorylation reaction is occurring. So essentially now we have a phosphate on carbon 1 and carbon 6. So again, substrate fructose 6-phosphate, product fructose 1,6-biphosphate. This is a phosphorylation reaction. We are adding a phosphate. Again, this is involved through kinases. And then the proper name is kind of cut off here is phosphofructokinase or PFK. This is a really important step in a lot of metabolism stuff where certain levels of PFK can have downstream effects and change the way our metabolism works. But that's a little more advanced, and you probably will see that in more advanced biochemistry courses or nutrition. So again, step 3, fructose 6-phosphate. We need to add ADP, so this is a strong negative reaction. So it's spontaneous and irreversible using phosphofructokinase with a negative 14.2. So step 4. So step 4 is fructose 1,6-biphosphate. Again, this is the beginning of the splitting phase. So here we essentially have the split going into this. So again, this is one of the exceptions of a large number, but it is reversible. So fructose 6-phosphate becomes dihydroacetone phosphate or DHAP, and glycerol 3-phosphate, which you'll see as G3P quite often. So again, substrate fructose 1,6-biphosphate. The products are DHAP and glycerol 3 G3P. This is a lysis. So again, we talked about lysis being involved in splitting. So this splits the carbon to carbon. And this is also involved in isomerization. So it's changing shape and splitting. Again, this requires a lyase and a isomerase. And the name is aldolase. That's the proper enzyme name. So remember that a common name for lyase enzymes is aldolase. So again, here's the one exception. It has a large positive, so therefore it should be non-spontaneous in a reversal reaction, but it is one of the exceptions. So again, we use our aldolase, we have isomerization and lyasing at the same time. So the change of free energy with the aldolase reaction is step 4. So again, this is when we look at the Gibbs standard free energy, we assume 1 mole concentrations. The cellular concentrations are 1,000 times lower in the micro or nanomole level. So Gibbs standard free energy of aldolase is a positive 23 kilomole, and the Gibbs free energy of aldolase is actually negative 1.3. So it could actually be considered actually spontaneous and reversible. Since again now it's negative, so it actually is spontaneous, but it's also low, so it is reversible. So again, we kind of went over this is splitting reaction and our priming reactions. So we did step 1, step 1, 2, 3, 4, 5. 5 is kind of the same with the sense that you're getting a different molecule, but also this molecule as well. So step 5, we have our DHAP or dihydroxyacetone phosphate becoming glycerol G3P. So we start with our DHAP. Product is G3P. Again, this is isomerization. So you can see here this is essentially changing. And the enzyme requires isomerase. It is called triose phosphate isomerase. So DHAP and G3P phosphate are both trioses, which are monosaccharides with 3 carbons. So this is when we split into that from 6 to 3. 6 to 3 carbons. So there's our 1, 2, 3. 1, 2, 3. And so plus 1 phosphate. So again, we have 1 phosphate. So it can be named triose phosphate. Again, relatively small positive, so it is spontaneous reversible reaction. Step 6. So step 6 is G3P to 1,3-biphosphoglycerate. So again, the numbers locate the carbons. So again, there's 2 biochemical processes involved in this reaction. So we go from G3P, and we have something actually added in here, NAD and a phosphate. So here's an NAD and a phosphate. Then our biochemical product is 1,3-biphosphoglycerate and an NADH. So that's quite important because we talked about electrons. So again, this is a phosphorylation and dehydrogenation reaction. So dehydrogenation involved in our NAD to NADH, and a phosphorylation with our G3P to biphosphoglycerate. We require a kinase and a dehydrogenase for oxidation. And the possible enzyme name here would be glyceraldehyde 3-phosphate dehydrogenase. But in this case, the possible name is actually the name of the enzyme. So again, G3P into 1,3-biphosphate. Even though there is both oxidation and phosphorylation occurring, the enzyme name will be named for the oxidization reaction, hence dehydrogenase. So step 7, we go through 1,3-biphosphoglycerate to 3-phosphoglycerate. So this is, again, you can see here we're having dephosphorylation, so we go from bi to just phospho. We go from 1,3 to just 3. So again, products of phosphoglycerate. This is a dephosphorylating. Again, that dephosphorylation involves phosphatases, and the enzyme name is 3-phosphoglycerate kinase. Again, the enzyme is always named for the reaction in which ATP goes in, rather than the direction in which ATP goes. So this is one of the reactions where we actually essentially starting the process of getting ATP. So again, this is known as BPG. This is 3-BGA. So again, this is a large negative, but this is actually one of the ones that is an exception where it is reversible. You can see the double arrows. And this uses a phosphoglycerate kinase. It should be spontaneously reversible, but it's one of the exceptions. So step 8. Whoops. Sorry. So step 8 goes from 3-phosphoglycerate to 2-phosphoglycerate. So again, we can see this number changing. So again, that's important. So when we see the same molecule, but something's changed, we can see that the phosphate is moved from 3 to 2, So again, it's changing shape. This involves an isomerization. But in this case, it's called a mutase. But we'll talk about that soon. So again, we go from 3-phosphoglycerate to 2-phosphoglycerate through isomerization. The enzyme required is a mutase, which involves the movement of a phosphate group. So even though it's isomerization, it's changing of structure, if a phosphate is being moved, this involves mutase enzymes. So we can see here the phosphate is moving from here to up here. And then the hydrogen is going down. So the enzyme is 3-phosphoglycerate mutase. So remember that mutase enzymes catalyze internal transfer of phosphate groups. So again, phosphoglycerate mutase is reaction of step 8. Again, this is a small positive reaction, so it should be therefore be spontaneous and reversible, which it is. So step 9, we go from that 2-phosphoglycerate into phosphophenyl pyruvate. So we can see that this might be changed once more. So here we can see one change where we actually see the removal of water. So here we see our OH. Our OH, and then here we can see it's being removed. So here's our H, 2 H's, and a 1 O. Now we can see the H is gone, as well as the OH. So we go from 2-phosphoglycerate to phosphophenyl pyruvate. This is a dehydrogenation reaction and involves a lyase. So this involves a dehydratase and a lyase, and the enzyme is called enolase. So here we see enolase involved in this dehydration and lyase reaction. So we see the OH2O being removed. And another name for enolase is 2-phosphoglycerate hydrolase. So again, small reaction, spontaneous and reversible. Step 10, the final step of glycolysis, is phosphophenyl pyruvate into, which you also see as PEP sometimes, into pyruvate. So here we have another example where we're generating ATP. Large negative reaction, so spontaneous and irreversible. So we can see another example involves dephosphorylation in a kinase. So we can see the phosphate is being removed and we're actually gaining that ATP. So again, phosphophenyl pyruvate becomes pyruvate, and remember there's 2 times 2 pyruvates. They require a phosphatase, but is named a kinase. Because again, we follow the ATP. The name is pyruvate kinase. Sorry. So remember, because enzymes are named for the reaction in which ATP is used in the forward direction, this is the enzyme kinase. The kinase enzyme even is named a kinase enzyme even though it removed the phosphate. So this is why it's a kinase and not a dephosphorylase. So again, large negative change of free energy and is spontaneous and irreversible. Oops. So everything we just talked about it can essentially be summarized in this figure. So we kind of just chunked everything out, each one individually. So again, if you want to just honestly draw it out a bunch of times, that's honestly the best way to get it down, is just draw it. And then you can also, I recommend that you draw it a bunch of times, but also do it this way. So maybe make a table and write substrate, product, process, enzyme name, maybe enzyme required. And then you can kind of write this 10 times and then slowly fill it in. And then you have all the processes down, and then also draw it to get the flow, and especially get down the things where ATP is used, ATP is gained, when we get our NADH, and water. So those are key steps in understanding where, and those things aren't just destroyed or gone forever. They have a purpose. So it's really important to know where they go from, because they can be recycled. They go to other parts of the pathways later in cellular respiration. So keep that in mind. So again, remember that glucose has 6 carbons and pyruvate only has 3. This means that 2 pyruvates are formed in each glucose. So we went through this pathway and followed this, but again, this is happening twice. So it's 2 pyruvates, and then each one of these would individually enter the citric acid cycle. So again, that's the end of glycolysis part 1 in cellular respiration. Hope you guys enjoyed it. It was a little long, but again, Just go through it slowly and just redraw it out and rewrite it out, and you'll get it. It's just one of those things you just have to keep practicing. It might fade slowly, especially like some of the enzyme names, but you want to keep those big key parts where I went through that one table where it's the 3 stages. I think that's a really good study figure. And then if you want the more detail, write out all the steps and draw it out. See you next time.